Going to talk today about next generation genomics for rare disease diagnostics. Okay, and here's a quick overview of what we're going to be discussing today. So first, we're going to be discussing Myome's whole genome platform and how it's really designed to meet patients' evolving diagnostic needs. Next, Dr. Danny Miller will be speaking about his leading work in the space of long-read sequencing and methylation. And then finally, we will end with Myome's head of clinical affairs, Lindsay Myers, to give a brief look at what's next here at Myome, focusing on our active research and actually how we might get some help from you to make some of these next steps happen in rare disease genomics. All right. So at Myome, I would say our mission is really simple but powerful. It's really just to deliver clinically relevant, personalized genomic insights on demand, integrated seamlessly into a person's health throughout their life. Our platform is built to make genomic information actionable, not just at one point in time, but continuously as science and technology evolve. And while Myome may be a new name to some people in this room or this webinar, this company has really been a labor of love for several years since its inception. Myome was built with the support of many visionary investors. You can see some of these names on the left. Some world-class commercial partnerships such as Natera, Broad Clinical Labs, and Illumina, and a growing network of research collaborators. And we're really thankful for all of these people's help to really make this technology accessible to our patients and families. This foundation allows us to translate some of this cutting-edge science into scalable clinical solutions. And we want to take a step back first and just look at today's testing reality. So families often go through a long, exhausting diagnostic journey, starting with microarray, sometimes moving to gene panels, exome sequencing, sometimes multiple reanalyses, before they even ever reach a genome test. Each one of these steps can add months or even years of uncertainty to a patient's journey, and we know that an earlier diagnosis can lead to more timely treatments, things like fewer unnecessary interventions, reduced costs for both the patient and the healthcare system, and importantly, identify resources for the patient and family just for support. Oops. And fortunately, the field is moving forward in this way. So as many here likely know, leading medical societies such as ACMG, NSGC, and more recently, the American Academy of Pediatrics, or AAP, now recommend exome or genome sequencing as first-line tests for many pediatric conditions. Some of them listed here: developmental delay, intellectual disability, multiple congenital anomalies, and epilepsy. This is really important because according to the literature, genome and exome have superior diagnostic rates than some of those traditional methods like CMA or targeted panels. And when looking at intellectual disability, developmental delay, and epilepsy, for example, genome sequencing can actually be shown to identify over 5 times more diagnoses in patients. So this evidence really just underscores this new standard. So comprehensive testing up front leads to faster and more complete answers for families. At Myome, we have built our diagnostic platform around a whole genome backbone for every patient. So what this means is that even when a clinician orders a copy number analysis, for example, or exome level test, we're generating data at a genome level quality. And this provides a few benefits for patients and providers. So one, using the PCR-free 30x genome as the backbone for testing, this allows for more uniform coverage and improves variant detection even for exonic regions. And this also means that there's improved CNV detection and the ability to detect tandem repeat expansion disorders, which we'll talk about. Secondly, once a patient is on the Myome platform, this allows for easy re-requisitioning of data without the need for additional samples, even as our knowledge or the patient's clinical picture evolves. And finally, this approach allows for adaptability. So the ability to integrate new technologies such as the ones we'll hear about today. And this approach ensures that every patient really benefits from that full genome resolution, and every test can evolve as our technology advances. So one example of this, as I mentioned, is related to tandem repeat expansion or TREs. And we know that these conditions are historically difficult regions to resolve using standard short read sequencing. So these typically require additional confirmatory methods to properly identify those repeat numbers, which are essential for making a diagnosis. So earlier this year, we expanded our platform to allow for TRE detection, and you can see our approach here on the right. So as always, we begin with our whole genome sequencing platform. And even using the short read technology, we can identify signatures that would indicate a potential TRE. And in these cases, we then move to our long read sequencing detection platform for secondary confirmation and to resolve that repeat number. Right now, we offer TRE analysis for 20 different TREs, and it is automatically incorporated into genome and exome orders. And Lindsay will speak how we hope to expand this list and see how we can use your help in doing so. Oops. Now, just very briefly, we wanted to cover our rare disease product portfolio. We do have a proactive arm of our business that we're not going to go into today, but using that whole genome backbone, we have these three different analyses that we can offer to patients and families. So, of course, that genome analysis with many benefits underneath listed, exome analysis with the benefit of that genome resolution, and then copy number analysis with the option to add Fragile X to this using that TRE platform. And this is available for proband only, duo, and trios. As we know, as we incorporate more familial samples, this helps us with that interpretation of the genomic sequence. Okay. And finally, we are consistently expanding the capabilities of our platform at Myome. So earlier this year, as I mentioned, we introduced that confirmatory long read sequencing to better capture those tandem repeat expansions. And we also added mitochondrial genome analysis to further expand that diagnostic power. Looking to the future on our roadmap, the goal is really to provide primary whole genome long read sequencing, the benefits of which you'll hear from very shortly. And then we are also investing in things like deep phenotyping and automated reanalysis, ensuring that every genome remains clinically useful as our knowledge grows. So in short, this platform really is not static. It's really built to evolve with the science. Okay. And with that, I would like to introduce our esteemed guest, Dr. Danny Miller. Can we see this? Okay. He is an assistant professor in the Department of Pediatrics, Division of Genetic Medicine, and the Department of Laboratory Medicine and Pathology at the University of Washington. He's an attending physician at Seattle Children's Hospital, and his lab is developing long-read sequencing-based genetic tests with a goal of increasing the rate of genetic diagnoses, reducing the time to make a genetic diagnosis, and lowering those barriers to obtaining comprehensive clinical testing. So I'll hand it over to Danny.
Thank you, Renee. I appreciate the introduction, and that was nice background for what I'll talk about, which is primarily using long reads to evaluate methylation, but also a little bit of structural variation and other things sprinkled in because that stuff is fun to look at. Just a quick disclosure slide. So I have a research lab, as Renee mentioned. I see kids in clinic about 20% of the time, and then the rest of my time is spent on the research side. Our lab really thinks about kind of like what Renee was talking about, which is how do we simplify the overall diagnostic process? Right now, it's in many cases it's stepwise. This has, of course, changed recently with fewer and fewer panels being sent, for example, more copy number analysis being done on the exome or genome side. But we do still definitely see kids, families who have kind of gone through this extensive workup. And at the end, shockingly, less than half of them are diagnosed by either genome or exome sequencing. So we always, in the lab, we think about why is this and what can we do to improve this overall workflow and increase the diagnostic rate. We believe, and we think we have data to show, and others are showing, that using long reads as a single test can both improve the rate of genetic diagnoses, so get us likely over that 50% number, but also just simplify the overall workup and evaluation. So instead of sending multiple tests over multiple months, multiple years. We can use a single test collected at one time and get kind of all of the answers we need from that one data source. I have the 6 year, which used to mean kind of 6 months. I now think of it more as, 6 days, 6 weeks kind of thing. But we do know that it is possible for this to be something more like 6 hours to get these results back. So we see long reads really pushing the time to diagnosis down. So, what is long read sequencing and why are we so excited by it? I think the easiest way to think about long reads is to think about a challenging region of the genome that it can provide better evaluation of. And most often, those are repetitive regions. This is a very simple example where you have 5 repeats. It's just all CGG, which is pretty straightforward. But if you think about it, short reads, while they can evaluate the unique junctions between the repeat and the unique sequence, they have a difficult time actually telling you exactly how many repeats are here in some cases, and they have an even more difficult time telling you if there are any relevant sequence interruptions within the repeat. These can be clinically relevant, so FMR1, Huntington, etc. It can give you information about how likely it is for that repeat to expand, or it can give you information about how likely it is that that repeat, the penetrance of that particular repeat. With long reads, what you're doing is you're getting reads that are essentially as long as the length of the repetitive segment itself. So it makes the overall analysis of the region easier, and it kind of takes away the uncertainty about, well, how many units are here, or what's the actual sequence of those units. Interestingly, for repeats, it also is teaching us there's a lot of variability within each of us within these repeats, which has been fun to look at on the research side. If you want to put some numbers on it, for short reads, of course, I think everyone's pretty familiar with this. You're thinking of reads that are, say, 100 to 300 base pairs. They're highly accurate, and the cost to sequence to generate the data is quite low. It's under $500 essentially these days. Long reads, alternatively, are reads that are greater than 1,000 base pairs. They're variable accuracy, but we're seeing more and more where they're north of 99% accuracy on both the Nanopore and PacBio platforms. And then the cost to sequence a human to about 30x on both platforms keeps falling. We're now, I think, safely within the $1,000 to $2,000 range. And then, of course, recent announcements by PacBio have shown that there's maybe a $300 or $400 long read genome coming. But the key here, I would say, is that these are really information-rich reads. These are reads that contain not only the A's, G's, C's, and T's, but also contain the methylation information as well, which is really the thing that we're going to focus on today. So how is it that we can use long reads to evaluate multiple types of variants? Well, there's 4 big classes here. I'm not going to talk about each one of them today. I'm actually going to skip S and V and indels and just go straight to structural variants. This is kind of what got me into long reads early, and really it's kind of fun to think about still because there's a lot of things that we miss structural variant wise with short read sequencing that we can see with long read sequencing. So here's an example of some of the recent studies using short read sequencing and the number of structural variants that they find. And we see, shockingly, that they find less than half of the total structural variants that are present in any one of our genomes. So each one of us carries 25, 30,000 or so structural variants. If you sequence yourself with short reads, you're only going to find fewer than half of those. That's not to say that those structural variants that are missed aren't important. They are. It's just that we can't see them fully resolve them. So I'm going to give you a couple of examples of variants that were missed by typical testing and how we saw them with long reads. So this is an example of a kiddo who was born with a bleeding disorder. The clinical team suspected either afibrinogenemia or hypofibrinogenemia. He had standard clinical testing on an exome backbone. I think it was a panel on an exome backbone. It identified a single maternally inherited variant in the gene FGA, which, if you have the phenotype of a bleeding disorder, makes a little bit of sense because it's a fibrinogen gene. But there was no second variant found by clinical testing. So we did long read sequencing, and it's pretty obvious. We found this 4,000 or so base pair deletion. It removed the first exon and the 5' UTR of the gene. You can understand with exome sequencing how this might be missed. It's a small exon, and if you're just comparing coverage from exon 2, for example, to exon 1, you may not know with high confidence that there's less coverage here for this particular exon. The other thing, it should be obvious here, is that these are actually phased into haplotypes. We didn't need the parents to do the phasing. We're able to separate these reads just using the polymorphisms that are present in in all of us across each chromosome. So not only could we phase, we could see the deletion that was missed by standard short read sequencing. We've done this for a lot of individual cases, one-off cases, if you will, but we've also started to do it for larger cohorts of individuals. This is a really A meaningful example for me, this was a cohort of 8 individuals from 7 families who had a clinical diagnosis of Canavan disease, but they lacked a complete molecular diagnosis. Um, if you look at the pedigrees up here, you can see that across the 7 families, 4 of them had a single pathogenic variant found in the gene associated with the condition, which is ASPA, but no second variant. This is a recessive condition. 3 families actually had standard clinical testing and no variants were identified in the gene of interest. So we collected all these. We did long read sequencing of each one. And we found that all individuals who were missing a pathogenic variant, either one or both, had a single or had a 2,600 base pair insertion within an intron of ASPA. This is an SVA, so a sign allo VNTR, so a mobile element. And you can see with long reads down here on the bottom, it's relatively easy to see that, you see the read coming across, you see the large insertion, and the read continues. So you can see the insertion, you can also figure out its sequence. On top is whole genome sequencing data from one of whole short read whole genome sequencing data from one of those kiddos. And you can see that while the target site insertion is pretty obvious, where you see that bump in coverage, the insertion itself is a little hard to see. What you're looking for is split discordant reads or reads that aren't mapping properly. So it's not as obvious as it is with the long reads. It's not reliably called by structural variant callers in short read data. And when it is, it's called as a smaller insertion, maybe 400 or so base pairs. So with long reads, we can easily see the entire insertion and we can figure out its sequence as well. That was really important for this particular case because it lets us know, it gives us information about the pathogenicity of the insertion. So it contains a novel splice acceptor site that creates a new splice site, which is what ends up causing the gene not to be made. What was really surprising to us is how common this was. So first off, we were surprised that we found it in all of our unsolved cases in all 8 individuals. When we went and looked at Nomad and asked, well, how common is this variant compared to other pathogenic variants associated with this condition? We were really surprised to find that this is actually the most common pathogenic variant associated with Canavan disease. So the next most common variant is the single nucleotide polymorphism. Among our cohort, 3 individuals were heterozygous for this polymorphism. And you can see in our cohort, all 8 had at least one copy of this insertion, um, while 3 of them had 2 copies of the insertion. And we were able to use the short read data from Nomad to figure out that 66 individuals out of 120, or sorry, 66 haplotypes out of 126,000 haplotypes were heterozygous for this particular insertion. But again, it called it as a 400 or so base pair SVA. So with long reads, we got much better resolution of the insertion itself. With structural variants, there's kind of a problem of, how do you prioritize them? How do you know whether or not they're rare? Are they common? What's the allele frequency? How do you predict the impact of these? For the ASPA insertion, we did have the Nomad data that could help us determine the allele frequency. But for other events like the FGA deletion and other things that we see in the lab, we don't really have that information. So how do we generate that data? In our lab, in collaboration with Evan Eichler's lab here at UW and others across the country, we are doing long-read sequencing of samples from the 1,000 Genomes Project. This is a collection of 3,000 or so individuals, samples that were donated and are kind of open access for research, if you will, that can be used for identifying allele frequencies across populations. And we're using it specifically to identify the frequency of structural variants across populations. To date, we have sequenced about 500 individuals from this collection on the Nanopore platform, and there's more data coming on both the PacBio and the Nanopore platform. The thing we're most excited by is the coverage that we get, which is greater than 30x for each sample, and then the really long read lengths, which is greater than 40,000 base pairs for each sample. What this lets us do is it lets us build a nice catalog of structural variants that can be used for filtering. We've created tools to facilitate this. One of those tools is called Needler, created by a really talented grad student in the lab, Gus Gustafson. And you can see that one output of Needler is you just input your sample, and it will give you for any structural variants. For example, this deletion, it'll give you the allele frequency, which is 0.001. The gene, and it'll give you any OMIM association that goes with it. And this is an example of a family who had a hereditary breast and ovarian cancer and prostate cancer, but no pathogenic or candidate variant found by standard testing. So we think it's likely this deletion in this particular family. Needler's available on GitHub, and yeah, please feel free to reach out if you're interested in using it. The other really remarkable thing about structural variants is that filtering them using short reads is actually quite difficult, and it's actually a lot easier if you filter them using long read sequencing data. So this is data from 10 individuals with known pathogenic structural variants. We found them using long-read sequencing. You can see each individual except for one has about 20,000 structural variants per genome. The coverage for this individual is lower, which is why we see fewer structural variants. If we filter those structural variants using data from Nomad, which contains samples from over 120,000 haplotypes, we only filter out about two-thirds of the total structural variants. So about one-third remain. So 9,000 structural variants per genome. If you filter these structural variants using long read specific databases, only from, in this case, 480 individuals or nearly 1,000 haplotypes, you can see that we filter out more than 95% of the structural variants per person. So really filtering long read data using long read specific databases is key to accurately and efficiently prioritizing um, these variants. Just a real quick, what are we doing with this data in the future? So we're looking to generate data from all 3,000 samples from 1,000 genomes on both the Nanopore and PacBio platforms. And then if you're interested, we're also doing long-read RNA sequencing of more than 900 of the samples in collaboration with PacBio. So it'll be really interesting to see kind of the isoform diversity expression, things like that within this data set. So I talked about structural variants first for a very specific reason, because it feeds in very nicely to repeat expansions, which then feeds in very nicely to the methylation work. So when we think about repeats, the example I always like to use is from a study that was done, published back in 2019, here from the University of Washington. Heather Mefford was the lead author, or the senior author on this. It talked about two families with Baratelle-Scott syndrome. This is a skeletal dysplasia, and this is a recessive condition. And in both of these cases, they were unsolved, which we know the gene XYLT1. So the question was, what are we missing with standard testing? After a lot of work by Heather's lab, in both cases, she identified that the probands carried a repeat expansion within the 5' UTR of XYLT1. So this is a southern, here's one of the probands, mom and dad. You can see that the repeat length in mom and dad is below the wild type range, but in the proband, it's well above that wild type range. I should note that dad carries a deletion of this region, which is why the proband just has one copy of this repeat. Again, this took a lot of testing to find. So one question we asked is, could we basically recapitulate all of these tests using a single test using long-read sequencing? And the answer was yes, we could. So this is kind of our way of recapitulating the southern blot, where you see the proband has reads that have expanded compared to both mom and dad. Here's the gene, or the exon, first exon on the 5' UTR of the gene. Here's where that repeat expansion is. And you can see where the repeat expansion is, we get methylation and silencing of those particular reads. We see that in the proband here, which is shown by red. And then in mom, we see a whole lot of blue, which means it's unmethylated. But I will point out there's a couple of reads that are red, meaning they are methylated. And we think that's a premutation allele that gets methylated in mom. A lot of interesting, what would you do with that information type questions for premutation alleles, but I'll leave those to the side for now. But overall, what this showed us really was that we could use a single test, long read, to both evaluate repeat expansions, so identify the structural variant leading to the disease, but also have an explanation for why is the gene being silenced, right? It's being methylated by the expansion. So with that, I want to dive into what's hopefully the meat of the talk, which is what we're doing now with methylation and what I think we can do with methylation, which is pretty exciting. So I'm going to divide this into three sections, thinking about exon activation, thinking about imprinting conditions, and then thinking about epigenetic signatures. And I'll start with exon activation. And I'm going to start with data from the 1,000 Genomes Project. And I'm just going to show you kind of what we see when we start sequencing cell lines from XX individuals. You see a mix, if you can imagine, of either skewed or normal, if you will, or unskewed methylation. So what this plot shows you, so this is the X chromosome. This is one individual, and each dot represents the fraction of methylation at the CPG island. This isn't every CPG island. This is only a couple hundred CPG islands across the X chromosome. Not every CPG island is informative. What you see for each haplotype is you see basically a mix. So all the dots kind of, they average somewhere between 50, 40%, something like this. So this is what we expect when we see mixed exon activation. If we go look at IGV, we can see that in the reads actually. So these are phased reads. So haplotype 1 is up here, haplotype 2 is down here at the bottom. This is FMR1, here's the CPG island. And we can see that for both haplotypes, you have both blue, meaning unmethylated, and red, meaning methylated reads within the region. So this is what we'd see. This is mixed exon activation. We can also see skewed X inactivation, and this is what it looks like. So this is a separate sample, and you can see most of the dots either fall somewhere between, say, 0 and 10%, and the other dots fall somewhere above 75%. So you can see a clear separation of the dots. I should point out that the color changes just because the phasing changes. We can't fully phase these chromosomes, so the phasing is random. So going from blue to red really doesn't mean anything except that we weren't able to phase across this region here. Now, with by looking at this chromosome-wide skewing pattern, we can actually go look at the reads themselves. And again, we see what we expect. So we're at FMR1 again, we're in the 5' UTR. We see the CPG island. We see the two haplotypes, haplotype 1, haplotype 2. And you can see that haplotype 2 is completely inactivated and haplotype 1 is completely active. It's blue. Now, I don't think this is because there's a pathogenic variant here in haplotype 2. I think this is because for these 1,000 genome samples, these are LCL lines, these are cell lines. So, as they're immortalized over time, as they're being passaged, there's just selection for a certain chromosome. So within 1,000 genomes, we see a lot of skewing. We see maybe 15, 20% of the cell lines have completely skewed X inactivation. That's not what we would see in the population, of course. But we can use skewed X inactivation to help us understand the impact of variants on the X chromosome. So the example I want to show where X inactivation patterns help us understand a particular variant comes from a paper that was just published this year in HGG Advances. This is a paper that describes three families who were suspected clinically to have a condition called incontinentia pigmenti. I'm going to say IP from now on because saying incontinentia pigmenti is a lot of words. They had negative clinical testing. And we'll talk about why it's probably negative for these families. But they all had features of this condition. This is a lot of text. I just want to say that females were affected with rashes. They had the hair phenotype, they had the nail phenotype, they had the teeth phenotype. But the females who were alive in the families, the XX individuals, likely the hypothesis was they were able to survive because they had skewed X inactivation. And the question was always what's being missed by clinical testing. So just really briefly to show the phenotype, this is kind of what we expect to see in this condition. Neonates get this characteristic rash at birth. This is what that rash looks like later in life as it resolves. They get nail dystrophy, weak nails, very flaky nails, for example, adontia, hypodontia, so teeth abnormalities, and then hair as well, which I'm not showing here. This is the table from Gene Reviews that talks about, how often do you find a pathogenic variant when you do clinical testing for this condition. So again, the gene is IKBKG, used to be called Nemo. I'm going to ignore the males for a while because there's a huge ascertainment bias here where most XY individuals who have variants in this gene don't survive. So within XX individuals, if you do testing, about 65%, you'll find they carry the common 11,000 base pair deletion within the gene. About 9% you can find based just from looking at the ASGCTs, the sequence analysis, and then a smaller fraction are CNVs. This only adds up to about 80%, right? So we're missing 20% of individuals or unable to diagnose about 20% of individuals using standard clinical testing. So that was the question coming into this project. And why is it that we have such a hard time diagnosing this condition molecularly? Well, it's because the gene, IKBKG, which sits right here next to G6PD, mostly lives within a very large segmental duplication. This is about 50,000 base pairs, and it is essentially identical to a nearby stretch, another segmental duplication over here. There's a pseudo gene within here. It contains exons 4 through 10 of the gene. So exons 1 through 3 sit in the unique region, whereas 4 through 10 sit in the non-unique or repetitive region. And the problem is, as you can imagine, using short reads or even other, even PCR and Sanger sequencing, it's hard to get unique mapping, unique amplification of this particular region. So we were contacted a couple of years ago by Monica Wojcik, a neonatologist at the Broad, who's a clinical geneticist as well. She had taken care of a family kiddo in the NICU who unfortunately passed away at about 2 weeks of life with a phenotype consistent with incontinentia pigmenti. The family had a long history of a clinical diagnosis of this condition, but had never been able to make a molecular diagnosis. I believe mom had an exome, had targeted testing for this condition. The proband here had targeted testing. Everything was negative. And you can see as you look at the pedigree kind of going back, 5 generations, multiple pregnancy losses, kind of things you would expect for an excellent condition. Family really wanted to know what's causing this, right? Because they were looking at doing at IVF, but you can't do IVF unless you know what you're selecting for. So we got a sample from the kiddo. and we did whole genome long read sequencing. Here I'm showing you the reads, haplotype 1, haplotype 2, and this is over IKB-kg. So here's the gene in the bottom, and then you can see G6PD, of course, as well. The common deletion is denoted by this blue box here. And hopefully what you see is that in haplotype 1, this kiddo has a deletion that takes out exons 8 through 10 of the gene. So it's smaller than the common deletion, but pretty easy to imagine that this is likely a pathogenic event, removing those exons. On the other chromosome, there was no pathogenic variants, no deletion, things like that. So it's nice that we could see the deletion, but can we get more information from the family or from additional analysis? And the answer was we could. So we got samples from three living family members, maternal grandmother, aunt, and then mom. All three individuals who had a clinical diagnosis of IP also carried the same deletion. So it was segregating with affected individuals, which was helpful. And then using methylation, we could kind of start to understand why it is that the proband was so severely affected compared to the other family members. So again, phased data, it's the same view I showed you before, where you have the deletion of exons 8 through 10. Over here with the CPG, instead of seeing skewed exon activation, meaning like the bottom haplotype is all blue and the top haplotype is all red, we see a mixed pattern. So this individual is expressing the copy of IKB-kg that contains the deletion in about half of the cells. This is blood-derived DNA. This probably helps explain why her phenotype was more severe than her other family members. We can actually go look at the other family members, look at methylation, and we can see that indeed they have completely inactivated the chromosome with the deletion. So here's mom, completely red on the chromosome with the deletion. Same thing for aunt and same thing for grandmother. So not only could we use long reads to find the deletion, we could use it to help understand why these three relatives were essentially not as severely affected as the proband. Now, why she didn't have skewed exon activation, I don't know yet. That's a really good question and something we continue to look into for this family. Similar to those plots for the 1,000 genomes, we can do chromosome-wide evaluation of exon activation. And we see that when we see skewed exon inactivation at one locus, we see it across the entire chromosome in the three surviving family members, which is, again, what we expect. But then for the proband, we see a mixed pattern across the X chromosome. So a really nice way to show we can see the deletion, which is challenging to see, but also use methylation to both understand why individuals are doing well or why individuals are more affected than others. And then I just want to show the second family because I think it's always fun to think about how skewed exon activation can work in an individual who's XXY. This was a separate family, kind of similar story, multiple individuals with clinical diagnosis of IP, but no definitive, no positive clinical testing. This was the proband, the XX individual. You can see the evidence of the rash on her legs, and then you can see kind of dystrophic nails as well, which is characteristic of the condition. We did long read sequencing. She surprisingly carried the common 11,600 base pair deletion associated with this gene, with this condition. We don't know why this was missed by clinical testing. We look at methylation patterns, we can see that she has skewing, so completely inactivated the chromosome with the deletion, but the one without the deletion is active, which is what we expect. And what was interesting is that she had a brother who was XXY, who had similar features, and our hypothesis was that we would see skewed exon activation in him, and that's exactly what we saw. So this is one of his haplotypes, this is the other one. You can see the deletion is the same that his sister carries. I want to point out that some of the reads got mapped to haplotype 1 because the sample wasn't great, so the reads weren't really long enough to completely phase the entire region. So we do have some reads with the deletion that ended up on haplotype 1, but we do see completely skewed exon activation in him. So he's kind of undergone the same process, which it's kind of neat to think about in an individual who has two X chromosomes and a Y chromosome. Okay, I'm going to switch over to imprinting conditions now and think about how we can use long reads and methylation to improve, simplify, streamline diagnostics for these conditions. And of course, the one that we always kind of think about first is Prader-Willi-Angelman syndrome. So as many people on the call probably know, is standard clinical testing for imprinting conditions is stepwise. So you do one test, and depending on the result, you may do another test, and depending on that result, you may do another test. It's time consuming. Clinical labs need to maintain competency for multiple tests. That's expensive. You need to keep people trained in order to do the test, things like that. So we've always wanted to know is could we use long reads as a single test to basically replace these assays to show that long reads are non-inferior to standard clinical testing. And I'm going to focus on Prader-Willi and Angelman today. We're working on Beckwith as well, but we've basically wrapped up the Prader-Willi and Angelman work. So this is work that was published earlier this year in the Journal of Molecular Diagnostics. We took 20 individuals who we knew had either Prader-Willi or Angelman syndrome. So we took DNA from those 20 individuals, and then we included negative controls as well. We did whole genome long-read sequencing, and our goal was to create a report that could be used by kind of a non-technical, someone who didn't have to get on the command line to do the analysis, a variant interpretation scientist type role. Look at that report and say, okay, can I use this single data source to determine whether or not this is Angelman, Prader-Willi, or neither? And we looked at 20 cases, and I'll say 3 independent people looked at these, and in all 20, all 3 people got it. For all 3 people, they got all 20 correct, which was great. So the way our report is laid out is we first showed the copy number within the 15Q region. And you can see the standard breakpoints here, which is where most CNVs occur in this condition. The dotted line or the dashed line is the genome-wide copy number average. You can see a loss in copy number, a loss in coverage between BP2 and 3, which would suggest a deletion. We can see loss of heterozygous polymorphisms. So heterozygous SNPs are plotted here. Homozygous SNPs are plotted on the bottom. So you see loss of heterozygosity. And then we can look at methylation within the region. And it really helps us kind of nail down the diagnosis in these cases. So you've got a deletion, you have loss of heterozygosity. And then if you look at the key CPG here in the SNRF-SNRPN gene, you can see that while your controls have about 50% methylation, which is what you expect, your affected individual here has basically no methylation. So that would suggest that this is they've got dad's copy of the gene, so they lost mom's or region. So this is Angelman syndrome caused by a deletion between BP2 and BP3. Again, for 20 cases, all 3 individuals got every one of these right, and they got the negative controls right as well. As most of you probably know, about 10% of Angelman cases are caused by pathogenic variants in UBE3A. Those are most often de novo. And there's always the challenge of phasing that de novo variant to figure out is on the maternal or paternal chromosome. Often we just rely on the clinical presentation, right? There's what presumably de novo variant in this gene. They have the phenotype, so we call it Angelman syndrome. What we wanted to know is could we actually phase across the entire region? So between UBE3A and the key CPG that's used to determine whether or not the chromosome came from mom and dad. So we had one case where we had a pathogenic variant in UBE3A. It's a 3 base pair in frame deletion, but this is pathogenic. It's on haplotype 2 down here. So we've phased the reads across this region. It's about 500,000 base pairs. And we can see at the key CPG that this deletion is on the chromosome, which is methylated. So this is on the maternal chromosome. So this confirms that this individual Angelman caused by a de novo variant UB3A, and it shows us that it is actually on the maternal chromosome. All right, so I'm going to switch gears one last time and talk about epigenetic signatures. This is a region that our lab is just starting to get into, but we're really excited by this because we think there's a lot of potential here for expanding diagnostic yields, but also helping clarify uncertain variants as well. So patterns of methylation genome-wide can actually be used can be associated with particular conditions. That's true for things like cancer, but it's also true for things like rare disease. So this is a figure from a paper published by Beckham Sadovic's group, who's been a real leader in this field, showing that for a particular condition, in this case, Cornelia de Lange syndrome, you can take a group of controls, which are in green, a group of affected individuals, which are in blue. You can do either supervised or unsupervised clustering methods to figure out is there some separation between the two for a certain set of CPGs. So that's what they're showing here is that everyone who's a control in green groups together and everyone in blue who's a case groups together. You can then use these groupings to help figure out whether or not uncertain variants, which are denoted by the 2 red bars here, do these individuals group with the cases, the cases over here in blue or the controls over here in green. So with the heat map, they're showing you that they do, so they separate. So you would say that this person, the VUS maybe is not pathogenic, whereas this VUS could be pathogenic because it groups with the cases. You can see the same thing on a PCA. At the separation, you kind of have to squint to believe that the line goes through it. The heat map looks nice. The PCA maybe not as nice for this particular example. But you can also develop scores. You can develop these SVM scores, which say, you know what, this individual who has this VUS, it scores or it groups really nicely or accurately with the folks who have Cornelia de Lange syndrome. This is really great and exciting, and I think there's a lot of potential here for the clinical genetics field. Unfortunately, much of the data and the methods that underlie these signatures isn't publicly available. So there's only a limited amount of signatures or conditions for which we can actually go in and look at the data, understand how the clustering or grouping is being done, and understand really some of the results we're getting back from this testing. So one thing my group is doing is we're developing publicly accessible methods and data sets for epigenetic signatures. This is work being led by another talented grad student in the lab, Sophie Gibson. What she's done here in this heat map is she's taken two sets of data, array-based methylation data and also long-read data. The array-based data is for Kabuki. You can see that we have a set of controls, which is in blue. The green denotes that this is array-based, and the red, or salmon maybe, I don't want to call it red, denotes that this is long-read data. And then over here, you have individuals with Kabuki syndrome. You have the array-based data, and then you have long-read data as well. The first question we had when we started this was, could we use the array-based data to group and cluster long-read data? We actually didn't know if this would work, but it does work. So here you'll see in the controls, we the array and the long read data group together, and then for the Kabuki case, the array and the long read data group together. We have more than one case now. When we made this plot, we just had the one, but we've had several additional Kabuki cases now. And they all group together like they should. This is the heat map. This is the PCA, again, showing you that we can actually separate the controls from the cases. Circles are array-based data, and the triangle is the long-read data. Again, they group and cluster very nicely. So we know that we can use array-based data to make these groupings. The next question is, could we build signatures using long-read data only? And the answer is we can. So this is unpublished data. This is our first effort to do condition-specific methylation classifiers. This is for Coffin-Siris syndrome, a bathopathy. And you can see that for all of our cases, the triangles are individuals with Coffin-Siris syndrome. You can see that they separate or group very nicely from controls. So this is all long-read data over here for control individuals. This is an important one for us because the array-based data doesn't seem to give as good a groupings, and there can be some overlap between cases and controls with Coffin-Siris. So we're really excited to see this really nice separation between these two. This is work that hopefully will be out, available online for people to use, and also out end of the year, early next year. I will say that we are all very excited in growing these databases of methylation signatures. So please, if you have families who you think would be interested in contributing a sample, or if you have unsolved cases with a compelling variant, please reach out, because we would love to take a look at them using long reads and see if we can group them into the appropriate condition. Okay, so I'm going to wrap up with just kind of a couple thoughts about, how do we really, what do we need to do to bring long reads into the clinical environment at scale? The first thing is that we need better, larger, more comprehensive databases of variation from diverse populations generated on long-read platforms. I showed you earlier that for structural variants, even if you have hundreds of thousands of genomes with short reads, you can't filter a sample as well unless you use long-read sequencing specific data. So that's true for SVs, it's true for repeat expansions as well. For methylation data, as I just talked about, we need to build databases that are accessible, methods that are accessible to everybody, open to everybody, for grouping conditions based on methylation signatures. That's ongoing, but it needs to continue. On the lab side, a couple of kind of boring things, if you will, need to be done. We need better methods for handling samples, for preparing libraries, for loading, running samples, things like that. The whole workflow just needs to get simpler. And I think everyone's starting to realize this. And I think the major sequencing companies and also robotics companies are seeing this. So we're seeing good progress there. We, of course, need really good tertiary analysis pipelines that use all aspects of long-read sequencing data. So that includes methylation, that includes phasing, that includes, being able to associate pathogenic SV with a single nucleotide variant, for example. We, of course, have to get payers to support this, to pay for it, which I think they're moving towards. And then we definitely need better resources for both pretest counseling for this complex testing, but also discussing more complex results. And the example I always give is that, you need more than 30 minutes to discuss a mosaic inversion in IKB-KG that causes incontinentia pigmenti with a family. 30 minutes just doesn't cut it. You need resources for that. So with that, I'll stop there. I'll say thank you to the lab for the work that goes into this. We have lots of collaborators, not all of whom are listed here, but they really help make this work possible. And then thanks to Myome for hosting this webinar. And I will stop and give it back to Renee and Lindsay.
Awesome. Thank you so much. I am just going to reshare and hand it over to Lindsay to wrap up.
Thanks. And I'll be quick here, but wanted to touch on some of the things, research and validation efforts that we have going on at Myome. And thanks, Dr. Miller, for your presentation. I always appreciate listening to you and the way you can break down some of these more complex things in a way that I can understand and enjoyable to listen to. So always a pleasure listening to you. But covering some or looping back to some of the things that Danny were talking about with improving detection rates using whole genome long read sequencing is where Myome wants to be in the future. As Renee already kind of alluded to earlier, we have now incorporated targeted long read sequencing for confirmation of our TREs. But in the future, we want to broaden the use of whole genome long read sequencing, including methylation signatures, so that we can kind of bundle these together with the short read sequencing to improve detection rates. And so with that, some of the efforts that we currently have going on in our validation is one, we're validating methylation signatures for a group of methylation-related disorders. And we too are looking for samples that could help expand the group of conditions that we are validating. So the more conditions that we can validate, the more that we can then in turn offer to you commercially so that you can order these for your patients and help expand, diagnoses for patients who maybe are clinically concerned or in the differential, but testing has been thus far inconclusive, or helping with VUS resolution as well. And so that's where we need your help. We are seeking patients who have molecularly confirmed methylation-related disorders to partner with you to help grow our data set so that we can broaden our commercial offering. So if you have families that are affected by these disorders or know of resources that you would like to connect with us, we would love to collaborate. What I'm showing here on the right side is a patient-facing flyer that we have that outlines kind of this validation research study that we have. We do compensate patients for their participation and providing us with a sample. And we do have a QR code on this flyer that we can provide to interested individuals that links to a page that lists all the conditions we're currently targeting. And our team would handle all aspects of consent, sample collection, and participant compensation. So if you go to the next slide. Here, yep. So, sorry, the one before. I was looking at the wrong screen. So this is a list of the conditions we're currently targeting for methylation. We are also looking for samples for those with tandem repeat expansion disorders that we don't currently include in our 20 genes, so that we can also expand that offering. And this is also listed on this QR code, which we can provide in our patient handout. Right. And then you can go to the next one. And the last thing that I want to briefly touch on is our Rare Insights program. So we're working on bringing whole genome long read sequencing to be commercially available in the near future, but we recognize that currently there are families that could benefit from this now. And so to kind of bridge that gap, we have a program here where if patients have had undiagnostic testing through Myome, they can apply for our Rare Insights program, which is doing long read sequencing on a research basis. So you can see that participation criteria there listed on the right. This is really designed for patients who have one variant in autosomal recessive condition that strongly matches their clinical picture, or if they have no variants identified and their clinical picture is highly suggestive of a monogenic condition. So this is available to anyone who has had diagnostic testing through Myome. Providers that order testing from us can apply. There's a short form that we have, and we also proactively identify cases that come through us as well. We review each case internally, including Dr. Miller as part of this review, and then we also help coordinate consent and sample collection if a family we feel would be a good fit for this program. Okay, and that is the next one. And on that note, I'll wrap up. So we do have time for a few questions. But we wanted to say that we will be at NSGC next week, so please come visit us. We'll have our handouts on the Rare Insights program and also validation samples, flyers with that QR code that links to all of the conditions that we're currently targeting. And we'd love to talk just more about how our testing may fit in with your clinical practice. So both Renee and I will be there next week, so please stop by and say hi. But with there, we'll wrap up. Thanks everyone for joining. We'll answer some questions, but we're excited what's next for Myome and for the families we serve.
All right, thanks, Lindsay. So yeah, we have a few minutes left for some questions, so we can just go through what we've received in the Q&A. And anything that we don't get to today, I'll have Dorisha, if you could just throw our emails maybe in the chat or something like that. That way you can reach out to us, and the clinical team is happy to answer them. So one of the first questions we have is, this one is for Dr. Miller. In the repeat expansion case study where the long read analysis was able to replicate the previous result, how much of that positive result was influenced by already knowing the repeat expansion location in the genome? Would the results be replicated with long read analysis if the case was blinded?
Yeah, great question. Like, how did we know where to look?
Mhm.
Pretty easy when you know here's the positive result. I'm just trying to replicate it. But I think everyone is actively trying to figure out the best way to evaluate repeats genome wide. I'd say the easiest way to do that is if you look at the 70 or so repeats we know are associated with a disease right now, and that's typically the approach we take. Look at those, and then go to an expanded list, right? So, only look at CGG repeats and other genes, only look at 5' UTRs, kind of limit your analysis, but do it in a stepwise way. There is not a easy answer to your question. It's just to kind of do it methodically. But we will get there with genes that aren't associated with Omen phenotypes, for example. That will come.
All right, thank you. And our next question, where are long-read sequencing tests available to order clinically? I have a case of a patient with a VUS and an X-linked gene, and I'm trying to get X inactivation analysis, but it's been challenging due to financial barriers. I'll hand this over to you, Dr. Miller, first, maybe on a research basis. But I know that we did cover at Myome, we are working towards that clinical availability of long read eventually within the next year or so. But we do have that kind of confirmation piece right now available. And then Lindsay just spoke to our research offerings at Myome. Anything else you want to add, Dr. Miller?
No, I'd say Rare Insights might be a good.
Yeah. Great. Okay. How can interested physicians participate in Myome research? Well, Lindsay covered that. So I think Dorisha put in the chat, if you have any questions about getting patients involved in research, you can just send us an email at clinical@myome.com, and we can answer those questions directly on a patient-by-patient basis. Okay, next question. Do you have good resources that you reference or recommend for clinicians asking about the pros, cons, risks, benefits of moving forward with whole exome or whole genome sequencing versus CNA? Yeah, this is a great question. So we have a lot of Myome specific resources that we are happy to share, but we would love to work with anyone who has specific interests, and we're constantly getting requests to build out informational, educational resources for patients that are less kind of product driven, but really just educational in nature. So happy to, again, take any requests at Clinical and Myome, and we can take it offline. All right. So I know we only have a couple questions left, or a couple minutes left. So I think with no questions in the chat, as long as we've answered all of them, I think we can wrap up. So we just want to thank everyone for being here. Thank you to Dr. Danny Miller for the great presentation. Thanks to Lindsay. And yeah, feel free to reach out and come see us at NSGC.
Thank you, everybody.
Thank you.
Thank you.